Hello students, welcome to EPG Pathshala. I am Vidya-mitra. I worked as a professor of Biophysics at All India Institute of Medical Sciences. I was at JP Institute of Information Technology as a professor of Bioinformatics and at Dr. D.Y. Patil Biotechnology and Bioinformatics Institute as professor of Bioinformatics and director. I have been teaching biophysics for the last 45 years or more. Today, we are going to discuss about a module, structure of tRNA and modified nucleotides under the paper Biomolecules and their interaction. One of the inherent properties of RNA molecule is alternative folding, which can be a blessing or a problem. Post-transcriptional modification of nucleotide bases is a way to avoid any unwanted folding. Modified nucleotides occur in all classes of RNAs and have a great chemical diversity. Dear students, first let us see what we are going to learn in this particular module. First of all, I want to introduce you to what are modified nucleotides. This is very important. Next, I want to tell you the importance of modified nucleotides in tRNA structure. Then, we want to go through the details and intricacies in the structure of tRNA and the role of these structural details in tRNA function. Later, I would like to introduce you to the structure of ribosomal subunit. You know what is the ultimate objective of learning genomics and DNA structure and everything? Ultimately, we all want to understand how the proteins are synthesized. So, structure of the ribosomal subunit is one of the most important things. Alternative folding of RNA and modified nucleotides. Modified nucleotides are important for avoiding unwanted folding. From biosynthetic point of view, there are more than 100 base modifications known in tRNA. Modified nucleotides are used in several processes such as DNA-RNA labeling, capture separation, capping, nucleus stability, chain termination, linker modification, etc. And they have great commercial value and applications. Biosynthesis of modified nucleotides. Some are synthesized using simple chemical transformations such as addition of a methyl group. Example, methyl cytosine or thymine, or bond isomerization of uridine to yield pseudouridine. Others as dihydrouridine in redox reaction or uridine have a complex multi-step process involving action of several enzymes, and that too in a definite order to get these modifications. Base modification and physical properties. Modification in chemical structure leads to conformational and structural change in nucleotide and alter their properties. For example, methylation in M5C and T improves stacking because of its hydrophobic nature. Modification introduces structural changes by increasing steric encumbrance or blocking hydrogen bonding position at Watson-Crick positions. We show here in this picture number of modified nucleobases which are found in tRNA. For example, we have dihydrouridine or ionosine. In the case of ionosine, we have NH group and O6, but there is no amino group at the second position like in the case of guanine. For example, we show 3-methylcytidine. In this case, at the third nitrogen, which is normally involved for hydrogen bonding, we have methyl group attached. Pseudouridine, and in the case of pseudouridine, it is the CH which is really involved in connecting with the sugar group. There are several other modifications like N7-methylguanosine, lysidine, and so on. What do these nucleobase modifications do in the interactions? The stabilization effect in pseudouridine occurs due to base stacking and addition of hydrogen bond donor. Methylation at the 2'-hydroxy in sugar favors a 3' endoconformation of the ribose and blocks sugar-H interaction, which changes the hydration sphere around the oxygen. It also increases stability against hydrolysis by bases and nucleases. Change in the interaction. There are still more changes in the interactions. Modifications may also change the acceptor-donor pattern of the bases. A typical example is 3-methylcytosine. The formation of M1A blocks Watson-Crick position in adenosine and introduces a positive charge to the nucleoside M1A and M7G. At University of Albany, they are maintaining RNA modification database. In that database, there are 109 base modifications in RNA. Out of those 109, 93 were found in tRNA. 31 base modifications were found in rRNA and 13 base modifications were found in mRNA. Other RNA species had 14 modifications. Many modification motifs and their sequence locations are found to be conserved. This is very, very interesting. tRNA structure. The structure of yeast phenylalanine tRNA was first reported in 1974 by Kim et al. All tRNA's molecules have polynucleotide chains 75 to 93 units long. The structure is characterized by 3 levels of organization. The primary, which is the nucleotide sequence, the secondary, helices, loops, and sheets, and tertiary, the 3D structure similar to protein structure. We show in this picture the clover leaf model of the sequence of tRNA, and side by side we show the 3D structure. We'll describe it in details in next couple of slides, but you can have a general look that this is CCA, terminus, variable loop, Anticodon arm, Anticodon loop, D arm, D loop, and aminoacyl stem. And the same is shown here: aminoacyl stem, Anticodon arm, and variable region, D arm and T arm. The general pattern of tRNA structure is described here. tRNA molecule contains minor variants of nucleobases in addition to A, U, G, and C. All of them can be arranged in cloverleaf pattern. These have 5' terminal phosphate group, an acceptor stem with CCA end sequence is 7 to 9 base pairs long and made by the base pairing Watson-Crick and non-Watson-Crick. Let us see the structure of acceptor stem in tRNA. The amino acid is loaded to the CCA tail to the OH group of 3' end of A through a covalent linkage of aminoacyl tRNA synthetase. In most prokaryotic and eukaryotic tRNAs, the CCA sequence is added during processing and therefore it does not appear in the tRNA gene. There are few exceptions. Some prokaryotes do have CCA sequence coded by the genes of tRNA. This sequence is very, very important for recognition of tRNA by enzymes and important for translation of the genetic code. T and D stems. The first anticodon base or vowel position is sometimes modified to inosine. The T arm is 4 to 5 base pair stem containing T size C, where psi is pseudouridine. The D arm is 4 to 6 base pair long and ends in the loop that often contains dihydrouridine or D. That is why it is called D arm. The overall shape of 4 stems and cloverleaf is L-shaped with overall length 70 angstrom and width 20 angstrom. The two ends are 80 angstroms away. We show here Anticodon arm and loop and the difference between yeast phenylalanine tRNA and yeast asparagine tRNA. The Anticodon arm is 6 base pair and contains Anticodon loop. The tRNA primary sequence contains anticodon in the reverse order since 3'-5' directionality is required to read mRNA from 5'-3'. We show here these two anticodon loops. In the first case, OMG 34 red, AD 35 green, AD 36 blue are flanking out and they interact. Now, in the second case, where ASP tRNA, PDB code of this structure is 2-TRA, the red is G34, green is U35, and blue is C36. So you can see how the Anticodon loop is there. Yeast phenylalanine tRNA 3D structure is one of the most studied structure. Here the L shape is composed of two nearly perpendicular RNA helices and T and D stems. The outer rim of L corner is occupied by T loop. Acceptor and T stems are stacked together to form a continuous 11 base pairs double helix. Yeast phenylalanine tRNA, the Anticodon and D stems are arranged in a similar way, but the two helices are not straight but have a 26 degrees kink between the two stems. The two arms are related by pseudodiode symmetry bisecting the L angle. Some nucleotides in the invariant position, while some are variable. Now let us see the tRNA backbone structure. The number of nucleotides in the stem and loop is generally constant, except for variable loop, which contains 4 to 5 nucleotides in most of the tRNAs and 13 to 21 nucleotides in class 2 RNA molecules. Most of the tertiary edge bonds are between constant bases always. Base stacking is one of the most dominant feature in tRNA structure. We see Watson-Crick hydrogen bonding in many base pairs, which are horizontal in the structure. There are clusters of base pairs in the hinge region of tRNA. Non-Watson and Crick base pairing. tRNA has number of non-Watson-Crick base pairs. For example, G4 and U69 is a vowel base pair. G15 and C48 is reversed Watson-Crick with both chains parallel, and methyl 1 A58 T54 is Hookstein in antiparallel orientation, as Watson-Crick site is blocked by methylation. M2G26 844 is longer than Watson-Crick base pair and is also significantly non-planar. We show in this picture number of unusual base pairing, some of which we have discussed earlier in the case of tRNA structure. For example, we have 3 bases having hydrogen bond with each other. We have non-Watson-Crick base pairing, which we shall discuss in the next slide. A typical example is G10, C25, G45 is a base pair triplet. A23, U12, A9 also a base pair triplet. G22, C13, M7, G46 again a base pair triplet. In these cases, one base pair like G10, C25 in the first sequence is a Watson-Crick base pairing, whereas other base is attached at O6 or G10 from the major group side. The base pair is at the junction of D stem and Anticodon loop, and this is responsible for 26 degrees kink between the two helices. The bases of one strand are between the bases of other strand. The sugar puckers. The sugar puckers in case of tRNA belong to both C3' endo and C2' endo families. The pp distance is not constant but varies between 5.9 to 7 angstrom. Change in pucker angle is noted at interaction sites involving A9, G19, M7G46 and M1A58. The pi term. In tRNA, the PO torsional angle changes with the change in backbone folding. Two riboses are connected by phosphodiester bond and point to opposite direction. This is known as a pi turn. In tRNA, the PO torsional angle change with the change in backbone folding. Let's know something more about the pi turns. There are three classes of pi turns in tRNA. Pi 1 turn around U47, pi 2 turn around G15, have turn angles around 30 to 90 degrees or plus SC and 150 to 180 degrees AP, and pi 3 turn, which is a normal turn seen around C32. We show in this picture the comparison between the two tRNA molecules, tRNA Phi and tRNA ASP. The first structure is by Hingereti. The PDB ID is 4TNA, and the second by Westhoff et al. 2TRA. You can notice very easily that there is practically no difference in the two structures in spite of having little difference in the length of the sequence. Both anticodon loop, the CCA end, D loop, variable loop, all exactly look identical in these two structures. Aminoacilation. Aminoacilation is a process by which an amino acyl group is added to a tRNA molecule. Each tRNA is aminoacylated by specific tRNA synthetase. Usually, there is a single amino acid synthetase for each amino acid, though there may be more than one tRNA involved. Charging of tRNA. The aminoacylation proceeds as amino acid plus ATP going to amino acyl AMP plus PP. Then amino acyl AMP plus tRNA makes amino acyl tRNA plus AMP. Certain organisms can have one or more amino acyl tRNA synthetases missing in what is done at that time. In this case, there is a charging, and this leads to charging of tRNA by a chemically related amino acid. Now let us see the structure of a ribosomal subunit. Ribosomes are cellular organelles which make proteins from all amino acids. These are composed of RNA and proteins. On an average, mammalian cell can have 10 billion ribosomes. Ribosomes are composed of two subunits usually, a large subunit and a small subunit. These have usually short life. Structure of ribosomal subunit. Ribosomes attach to mRNA to form polysomes. After synthesis of polypeptide chain, the two units separate out. The subunits can be reused or broken up. As a result, small proteins can be made quickly. However, synthesis of large proteins takes several hours. Several physico-chemical studies have been used to yield the detailed overall shape of individual ribosomes and differences in the shapes for ribosomes from different species. Electron microscopy had been one of the most powerful techniques. Electron microscope reconstruction method has been used to study the interaction of ribosomes with mRNA, tRNA, and other protein elongation factors. Latter really helped for understanding atomic level details of the structure. One of the great discoveries of the present time is the structure of ribosomal subunits. In the year 2000, structural biologist Venkataraman Ramakrishnan, Thomas A. Stites, and Ada Yonath made the first structural subunit atomic level details available in the Protein Data Bank, PDB. These authors received Nobel Prize in 2009. Structures are also available for many other players in protein synthesis, including tRNA, elongation factors, etc. We show a beautiful picture of ribosomal subunit. This has been the picture of the year at RCSB PDB 2000. The two subunits of ribosomes are shown in this picture. These are composed of long strands of RNA, shown in these figures by orange and yellow colors. There are proteins. These proteins are colored blue. Ribosomal subunit structure. Let's discuss more. When synthesizing a new protein, the two subunits lock together with a messenger RNA trapped in the space between. The ribosome then walks down the messenger mRNA, 3 nucleotides at a time, building a new protein piece by piece. Now, there are several structures of the ribosomal subunits available at Protein Data Bank. We show in this picture a typical structure with the PDB ID 1FFK. The atomic level details can be seen on the figure on the right hand side, whereas overall view of the subunit can be seen in the left hand side. The large subunit. The large subunit contains active site of the ribosome, the site that creates new peptide bonds when proteins are synthesized. The mRNA runs horizontally in the groove across the middle. Enzymes typically use amino acids to catalyze a chemical reaction, but the ribosome uses an adenine RNA nucleotide, which was colored green in the previous slide, to catalyze the reaction. We show in this picture a small unit, which is ribosomal small unit. The structure of initiation like 70S ribosome complex with 8 base pair Shine Delgado SD helix sequence associated with the last part of RNA chain is shown in the subunit. The structure has been solved by Korostlev et al. In 2007. The PDB ID of this structure is 4V4J. The structure depicts how the Shine sequence lines up the mRNA in the right place, making it ready for a special initiator tRNA. The little piece of mRNA is shown in this picture in red, and tRNA is shown in yellow. The translation liberation screw analysis showed how inherent anisotropic motion of SD helix were biased along the helix axis. So students, what have we learned in this module? First, we tried to understand what are modified nucleotide bases present in tRNA. Then we detailed into the structure of tRNA. We saw the clover leaf model, the 3D structure, unusual base pairing, stacking of base pairing. Then we saw unusual turns, these are called pi turns, and sugar-Pucker backbone PP distances in the tRNA molecule. We tried to understand the mechanism of aminoacilation of tRNA. We saw the most important thing, the structure of a ribosomal subunit, large and small, and their function. Thank you very much.